The topic of permeability is an interesting one, and it's one that sometimes gives students a little bit of a headache at first when they're learning it, but it's a really, really critical topic because we refer back to this topic time and again in A&P 1 and A&P 2. So this is one of those topics that I really encourage you to make sure you get it down and understand it well, because you will be seeing this topic again and again. So let's go ahead and launch into it. And talk a little bit first about what crosses a plasma membrane. Now, I'd already mentioned that because we have a phospholipid bilayer that has hydrophilic heads and hydrophobic tails, it takes a very special kind of molecule that's able to even pass through the plasma membrane. Because that molecule has to be able to withstand both hydrophilic and hydrophobic environments. And I mentioned previously that molecules that can easily cross the plasma membrane tend to be very small, and they tend to be relatively uncharged. So for example, as I said on the previous lecture, oxygen, nitrogen, carbon dioxide, and water can all move across the plasma membrane fairly readily. It turns out that alcohols, small alcohols, also can. However, for the rest of molecules, things like sugars or proteins or amino acids, vitamins, lots of other ions and elements have to have some other route for getting across a plasma membrane. And that's where those transmembrane proteins come in. Because the plasma membrane does not allow just any old molecule to go across it, we say that the plasma membrane is selectively permeable. It's only permeable to a handful of items that I just mentioned here. One critical thing to know about the movement of molecules across any membrane: they always move from areas where they are in relatively high concentration to regions where they are in relatively low concentration. That is always true, and you need to know that. The natural movement of molecules is always from areas of high concentration to areas of low concentration. Now, anytime I have molecules moving from an area where they are in high concentration to an area where they are in low concentration, that by definition is a process called diffusion. Diffusion can happen in the air, diffusion can happen in a liquid. And this is really important, diffusion by definition does not need to have the molecules moving across a membrane. I'll give you a good example. If you stood on one side of the classroom and I stood on the other side of the classroom and I sprayed a bottle of perfume into the air, within a matter of minutes on the other side of the classroom, you would begin to be able to smell that perfume. The reason you'd be able to is because on my side of the room where the perfume molecules are in high concentration is different from the area of the room where you are, where they are in low concentration, and given a few minutes those perfume molecules move from me where they're in high concentration to you where they're in low concentration. Notice we didn't have to have a membrane between us in order for that diffusion to occur. So diffusion can occur in the presence of a membrane, and diffusion can occur without a membrane present. But the key is we say that the molecules move down their concentration gradient from where they're in high concentration to where they're in low concentration. Now, if we look at diffusion in a liquid, we are going to usually be talking about water with some sort of element or molecule dissolved in it, like salt, for example. If I have water and salt mixed together, I have what's called a solution. By definition, a solution is made of two items: a solvent, which commonly is water, although other liquids can also serve as solvents sometimes. And the other major component that makes up a solution is the solute. That's the thing that's dissolved in the water, such as in this case our table salt that we mentioned. Oh, I forgot to mention one other thing here. In this picture, we can show how if you put a crystal of dye into water, over a little bit of time, as all the molecules are moving into each other and knocking into each other, eventually you will get a uniform solution of the dye molecules and the water molecules together. And notice that the dye molecules moved from an area where they were in high concentration out into the water where they were in much lower concentration. That is diffusion. Now, if I talk about the movement of water molecules specifically, then I am talking about a special kind of diffusion called osmosis. But it still follows the rule of water molecules will move from areas where they are in high concentration to areas where they are in lower concentration. In other words, water also moves down its concentration gradient as well. We're going to talk about this a little bit more very shortly. Now, let's imagine that we put a cell into a solution. And let's imagine that that solution, let's see, let me draw this for you, that'll be better. Imagine we've got a beaker here with water in it, and I put a cell into that beaker. And let's say that inside that cell, the inside of that cell is 5% glucose, and remember that again, a solution is made of a solvent and a solute. In this case, the solute is glucose, the solvent would be water. So if we have 5% glucose, that means the solution is 95% water. Now imagine, let's on the other hand go out here and say, let's say the water that we put it in is 10% glucose. That means it's 90% water. Now, if we were going to try to predict what happens to the cell in terms of the movement of molecules across a plasma membrane, we would have to know a little bit more about how the plasma membrane behaves. Now, given what we know about the plasma membrane, we know, as I said before, the plasma membrane is selectively permeable, and only a small handful of items can go through the plasma membrane. Now let's just, for the sake of argument here, let's just imagine that glucose, even with the presence of transmembrane proteins, that glucose can't go across. Let's just imagine that. Glucose cannot cross the plasma membrane. Well, the problem with that is we still have one concentration inside of the cell here and a different concentration outside of the cell. And there's got to be something that occurs to try to equalize this. And as it turns out, if glucose cannot be what moves across the plasma membrane, water will. Remember, I told you water readily moves across the plasma membrane. So instead of glucose moving in or out of the cell to try to balance out the concentrations of solutions, it's going to be water that does the moving. So then the next question you have to ask yourself is, okay, which way is water going to move? Well, if you look at our little diagram here, we said that there is 95% water inside the cell and there's 90% water outside the cell. I told you that osmosis, movement of water, is always to move water down its concentration gradient from areas where it is in high concentration to areas where it is in lower concentration. So that means in this case, water is going to move from the inside of the cell outward because the water is in higher concentration inside the cell. And as water leaves the cell is going to begin to shrivel up or shrink up. Any solution that makes a cell shrink up or lose water is called a hypertonic solution. On the other hand, if on the other hand I were to place my cell into a solution that was 1% glucose, and 99% water, and here's my cell again, that's 5% glucose and 95% water. Again, we have a scenario where the concentration of glucose inside the cell is not the same as the concentration of glucose outside the cell. Again, assume glucose cannot move across the plasma membrane. If that is the case, the only other thing that makes up the glucose solution is water. water can move across a plasma membrane, and indeed it will move across a plasma membrane. In this case, it will move from where it's in high concentration of 99% to where it's in a lower concentration of 95%. So in this case, water will actually move into the cell to try to balance out the concentrations. That will cause the cell to expand or to swell up. We call that any solution that causes a cell to take on water, we call that solution a hypotonic solution. Now, what if we were to put a cell in a solution where the concentration inside and outside the cell of glucose was exactly the same? Let's say it was 5% glucose, 95% water, both inside and outside of the cell. So there's no need for water to move in and out of the cell to try to balance out any concentrations. And you might think, well, in that solution, then nothing happens, no movement occurs. But that's actually not true. Remember that all molecules are moving around and bumping into each other all the time. So there's always going to be some movement of water molecules into the cell, but it will be equally and oppositely balanced by water movement out of the cell. So yes, you do get movement into and out of the cell, but the movement is equal and opposite. Any solution where there is no net change in the shape of the cell is an isotonic solution. Think of that like an isosceles triangle is a triangle with two sides the same. An isotonic solution is where I have two solutions that have the same concentration. Now, in this last slide, what I'd like for you to do is see if you can figure out what would happen in each of these scenarios. So let me give you some numbers here. Let's decide that in this first cell, the inside is 2% glucose, and the outside here is 10% glucose. In the second one here, let's have this be 5% glucose. And that's 5%, and the outside will make it 1% glucose. In this one, let's make both the inside and the outside the same at 10%. Now, what I'd like for you to do is take a few minutes and actually draw yourself a picture and figure out in each of these scenarios what's going to happen with the cell. Hit pause right now on your recorder. Go ahead and do that. And then when you think you have the answers to all three scenarios, go ahead and hit play and check to see if you're right. Okay, so let's see how you did. In this first one, we had 2% glucose inside the cell. Remember, we assume that glucose cannot move across the plasma membrane. We have 98% water inside the cell. We have 90% water outside. That means if glucose cannot do the moving, then water has to do the moving. Water is going to move from where it's in higher concentration to where it's in lower concentration. In this case, water concentration is higher inside the cell, so water will move out of the cell, which makes this a hypertonic solution because the cell is going to shrink over time. Okay, in the second scenario, you have 5% glucose inside. That means it's 95% water inside. It's 99% water outside. Again, if glucose cannot move, then water has to be the molecule that moves across the plasma membrane. Water is in higher concentration outside at 99% compared to 95% inside, which means water is going to move into the cell, causing the cell to swell up. And any solution that causes a cell to swell, we call a hypotonic solution. The way I think of this is a hypotonic solution makes a cell swell up like a hippo. A hippotonic, a hypotonic solution. Okay, corny, but if it helps you remember it, use it. Last one, you have 10% glucose inside the cell, which means it's 90% water. Outside, it's also 90% water, which means Overall, there is no difference in concentration inside and outside of the cell. That means that overall we would not expect there to be any difference in terms of what happens to the shape of the cell. However, remember again, it's really important to keep in mind that equal numbers of water molecules will constantly be bouncing into and out of the cell. So there is still movement of water molecules. It's just that the net movement is zero. Any solution that has an equal concentration inside and outside of the cell is an isotonic solution. This is not an easy topic for people to get. You're going to have to practice it a few times to get it down. And as I said before, it's a really critical topic because we come back to it time and time and time again when we get on into A&P. And if you're going on to an allied health profession, these concepts become really critical for doing things like giving patients an IV that isn't going to damage their cells. So keep up the practice. Remember, you can always ask me questions, and I'll see you in part 3 of this lecture.